I've heard people say the word Friday about 15 times since I got in the classroom. So feel free to keep coloring. Well, it's not kindergarten, like I said, but it feels kind of like kindergarten. This activity is really important, though. It's going to come back either at the end of class today or on Monday when we meet again, when we continue talking about epigenetics. So it's just practice. Today we start talking about transcriptional regulation of genes. And so everybody should have read the chapter for bacterial gene expression or prokaryotic gene expression. And I just want to start with a simple question. Why is it necessary for organisms to regulate the expression of their genes? So let's imagine that you're a plant or an algae that can photosynthesize, like this one. So two different petri dishes. These organisms have been streaked out in patterns that look like the moon and the sun, I guess. If you're a photosynthetic organism, why would you want to regulate the transcription of your genes? What sorts of genes would you want to regulate? What genes do you not want to have turned on when it's dark out? Pardon? Right, sure. So genes that are involved in photosynthesis. So what's the point of wasting your cellular resources making proteins that are involved in producing energy from sunlight when there's no sunlight? So that's just one example of how gene regulation expression is important. That we don't make all of our proteins all the time. It's important to be able to turn the genes on when we need them and off when we don't need them. So in this video, which will work today. And by the way, there is no bird in here today. I checked. In fact, it wasn't in here a couple hours after last class, so it must have escaped. Yes.
Dr. Driver actually picked it up and threw it out.
Oh, did he really?
Yeah.
Good for him. I'll make sure we note that at our faculty meeting this afternoon. We'll give him a medal or something. So, what these scientists from Southern California were doing was they work with this algae, and they have some promoters. So here's a gene. The arrow indicates the position of the coding region. So you've got the promoter, which is 5' of the coding region. And promoters is where all the action is for gene expression regulation. Do you is transcription happening or is it not happening? That's all protein DNA binding in the promoter region of a gene, as we've already discussed. Right, this is where RNA polymerase binds, sigma factor binds, recruits RNA polymerase in the promoter. That's what causes transcription. In this algae, what they did was they took promoters that were, if I can spell it right, circadian rhythm promoters, kind of like you and I have, RHYTHM. One of the hardest words to spell. So they took circadian rhythm promoters. These are regions of DNA that drive cyclical transcription of genes. And they put those upstream of the gene for green fluorescent protein, or GFP. So these are transgenic organisms. They've artificially made this gene, green fluorescent protein, that's driven by promoters that turn on and turn off at different points during the day. And the only point of showing this is because I think it's cool, and I hope you think it's cool also. So this is a time-lapse video of these two petri dishes. The light goes off, so now they're just recording fluorescence. So over a 24-hour period, one promoter drives expression of GFP. That's why we see the light come on. It turns white. That's a green fluorescence captured by a camera that doesn't see green. It just sees light. So one of these plates, the green fluorescent protein gene is being driven by a promoter that turns on overnight. That's the one with the moon. And the other version of GFP is being driven or turned on. Its transcription is being activated right now by proteins that are only turned on, that only exist in the cells during the day. And these promoters are one way that this particular organism can sense light and translate that into whether or not certain genes are being turned on or turned off. So that's what we're here to learn about.
You're saying they turn off the light because they shine a light on the sun, so the one that looks like a sun or whatever.
So both of these plates are being photographed underneath a microscope. So there's no light in the room. These genes somehow, it's the magic of biology, these promoters know what time it is. So even in a black room, the promoter for this gene, the promoter that's driving GFP in this strain, is on for 12 hours overnight. And this is a daytime promoter. Thank you very much. That's the sort of feedback I love to have. Well, I'd rather have. We love it, it's amazing, but thank you very much. So, right, this plate has a GFP gene that's driven by a promoter that always activates transcription of whatever gene it's upstream of overnight in the dark. And then this plate has a different promoter. This is one that's on during the daytime.
So say like 8 p.m. to 8 a.m.
Something like that, yeah.
Wait, how are they testing that again?
How are they
testing that again?
They're not, so they already knew that these promoters had this activity. So they've just made synthetic genes to demonstrate the fact that these promoters turn on gene expression in non-overlapping periods of time. So the overnight promoter, they know, turns on other genes normally in these bacteria or algae overnight. And then when it's daytime, whatever these algae think are daytime, 8 a.m. to 8 p.m. maybe, that promoter turns on the transcription of genes that are involved in photosynthesis. Overnight, genes that aren't involved in the acquisition of sunlight, the capture of sunlight, get turned off. So somehow these algae sense. So what sort of senses do algae have? Or bacteria for that matter. Right, so they can sense chemicals. Does that taste or smell? Sure, I don't know. I'm being sort of playing fast and loose with my biology here. So the point of this is that every living organism, hopefully you all saw this in Biology 1, maybe, right? One of the properties of living things is that they can sense, that they react to their environment. And so even bacteria and algae sense. They have to have ways to detect what's going on around them and respond accordingly. Okay. So we have 5 senses. Maybe bacteria and algae have a sense for chemical, taste, or smell, whatever you want to call it. We know they don't have eyes, but somehow these blue-green algae sense light, despite not having eyes. So one of the things we're here today to figure out is, how do these unicellular organisms sense their environment, and how do they respond to the environment using transcriptional regulation? Yeah?
Is it maybe because of cell signaling, but I don't know, in like our psychology class, I heard that, I forgot what part of the brain, there's something that tells us that keeps our sense of time.
Yep.
We know when to wake up.
Do bacteria do that, though? No.
Cell signaling?
But they do, right, so in higher organisms that have multiple cells, then you get cell-cell communication. So you can have specialized cell types like light receptor cells, like our eyes, that tell us, oh, it's light out. Right. These unicellular organisms aren't quite that fancy, but they do still have molecules that sense light. And that's one of the ways that some of these organisms can regulate gene expression.
So it's not based on molecules that sense light at all?
In
the genes being on or off?
Not in the case of the blue-green algae that we started with. So that, was all done in a dark room. So there was no, they weren't turning the lights on for 12 hours and then turning the lights off for 12 hours and the algae were responding. The algae were doing that on their own. So like us, those algae have sort of built-in clocks. They sort of inherently know what time it is. We get tired. I don't know how I would describe what the blue-green algae do when they when it's getting late, but somehow their cells know to stop transcribing the daytime genes and to start transcribing the nighttime genes. Do algae have a sense of time? I suspect not. But they do chemical timing inside the cell.
Did they do the experiment with algae around the world, so because the time?
That would be cool. Fly some of the same species in from the other side of the world and then see if they do the same thing. No, I don't think they did, but that's a good, you should have been here. I think the people that did this work, the faculty member was here a year ago to do a seminar at our Friday afternoon seminar series. You should have been here because that would have been a great question to ask her. Other questions about this? So everything that's living senses. One of the most important things that bacteria can do in terms of sensing is to sense what sort of food they have around them. And bacteria can metabolize a lot of different forms of sugars, but they have different genes that help them metabolize those different types of sugars. Sucrose, glucose, fructose, and so forth. So if a bacterium is in the presence of glucose, they want to turn on their glucose metabolism genes, but they also don't want to have all of their galactose metabolism genes turned on, because if there's no galactose there, then what's the point of making genes that let them eat it? So that's one of the ways, and that's the case study. How many people, by the way, let's do a show of hands. How many people already learned about the Lac Operon at some point in biology? About half of you? Okay, that's what I figured, but the other half of you need to hear this as well, and we're going to go into a little bit more detail maybe than you've had in the past. So here's the promoter of a gene again. What facilitates transcription? How do we know whether or not transcription is turned on or not if we look at these two images? Bacterial cell. What's the gene that brings in RNA polymerase to start transcription? The sigma factor. So we have a sigma factor bound here to the promoter. It recruits RNA polymerase, transcription starts. Transcriptional regulation is all about proteins binding to DNA. Transcription itself is all about RNA polymerase binding to DNA and transcribing it. The ways that transcription is regulated, another way to say it is how is RNA polymerase regulated, is all about other proteins binding to DNA beyond RNA polymerase. So we have to talk about a couple other different types of proteins, activators and repressors. Activators activate transcription, repressors repress or turn off transcription. So the first critical thing to figure out is how do these proteins do those jobs? How is it that activators activate and repressors repress? So what do you think? It says down at the bottom of the slide if you don't want to think, but how would the binding of an activator protein like this here under positive regulation, how does the binding of an activator protein to part of the promoter help turn on transcription? What sort of things could that protein do that would make transcription occur more frequently or make it more likely that a gene gets transcribed? Let's say it physically interacts with sigma factor. So sigma factor binds in the promoter. This activator protein physically binds to sigma.
Very strong affinity to the sigma factor.
Right. So we've got two proteins that bind to each other. So you have a activator molecule that binds to DNA in the promoter. That brings in sigma factor, or keeps sigma factor, grabs onto sigma factor and holds it there at the promoter. Sigma factor binds to the promoter. Sigma factor then brings RNA polymerase. It gets it's very stepwise gene expression regulation. There's a lot of proteins that are involved in a lot of steps often that are involved really in understanding how transcriptional activation works. So that's one way that binding of a protein to DNA could help enhance or increase the amount of transcription. What about the other side? repressor protein binds to a DNA sequence. In this case, repressors bind to sequences that are called operators. It's a DNA sequence that's in the promoter. We just call any sequence a repressor protein binds to an operator. Apologies again for the terminology, but that's the way geneticists talk. The activator binds to an activator binding site. The repressor binds the operator. How does the repressor stop transcription? By changing the shape of what? I agree. So what happens to, where would RNA polymerase be in this drawing? So let's say sigma binds the promoter. Where would we put RNA polymerase? On top of the, yeah. So one way that repressors work is they physically prevent RNA polymerase because the repressor is bound to DNA in the promoter region. It physically prevents RNA polymerase from accessing the DNA or from moving from the promoter through the operator sequence into the coding region. Just like a roadblock. When repressors
are technically the binding site of the RNA polymerase then as well.
Well, not necessarily. So the question is the operator also the binding site for RNA polymerase? Not necessarily. So that's sort of case-by-case basis. Generally, sigma factor will recruit, let me be more specific. Generally, sigma factor would recruit RNA polymerase to the promoter, but the operator sequence happens to lie between the promoter and where transcription starts, where the arrow is there. So in this drawing, the way I've drawn it now, that repressor protein that's bound to the DNA is just preventing RNA polymerase from sliding across the DNA of the operator sequence into the coding sequence. So two ways repressors can work. They can either prevent RNA polymerase from binding to DNA in the first place, or they could prevent RNA polymerase from moving down the DNA and transcribing. Other questions? So proteins binding DNA can enhance transcription by helping stabilize RNA polymerase, that is, keep it on the DNA, or by recruiting other transcription factors, sigma factors, for example.
Let's say we're going through a period where it needs these proteins, and it makes the protein that it needs for that event.
Yep.
So it makes the protein that it needs for that event to go smoothly. Why would it want to do that again if it already made the proteins that need the proteins go away? Essentially, what happens to those proteins that are already made?
Fabulous. So the question is, what happens to proteins after they're made, and why do we need to regulate transcription once transcription happens, translation of those messenger RNA molecules happen, you've got all these proteins. What's the point of regulation at that point? So the critical question is, what happens to those proteins, as you put it? So the proteins are doing their jobs, and at some point the proteins degrade. So all proteins, like all messenger RNAs, at some point degrade. Sometimes, and a lot of times, and you'll learn more about this in cell bio probably, if you haven't taken it yet, is that there are cellular mechanisms that actually target proteins and destroy them at certain periods of time. So frequently, depends on the protein, but once a cell makes a protein, that protein doesn't stay in the cell forever. Eventually it gets degraded, turned back into amino acids essentially, and reused as building blocks to synthesize new proteins. That's why, and I should have pointed this out in the video of the glowing algae, that's why you don't see them glow forever once the green fluorescent protein is made. That's why when the transcription of those genes, the GFP genes, goes down, you don't see the strains of algae keep glowing. Because those proteins that are actually making the light that we see get destroyed. So in those poor algae, every day they have to turn on those genes and make all the same proteins over again. But at least they're not making them 24/7. It's a trade-off. Other questions? Proteins binding DNA can either improve, increase transcription, or not. That's just some terminology. Know the words in red. We've already gone over this. Okay. So here's the key point. How does this process respond to environmental conditions? How does the ability for proteins to bind DNA help an organism sense and respond to its environment? So in this case, this cartoon from one of the textbooks that I used previously, we're talking about one of these proteins. Let's say it's any sort of regulatory protein. It could be an activator or it could be a repressor. So in this case, the one on top here is shown as an activator. Most of these proteins have at least two different jobs they can do, two different functions. One is those proteins bind DNA, so they have a DNA binding site, part of the protein that physically interacts with the DNA sequence that it recognizes. What's an allosteric site? That's the other role of these proteins. Does anybody remember what the definition of allostery is?
Another site on the protein for another effector can get the same effect out of the protein.
Right, it binds something. It binds an effector. So an effector, yes, exactly, an inhibitor, or, well, the inhibitor is, terminology gets a little bit difficult here, so I try to avoid it at all costs. Those regulatory proteins in their allosteric site bind a molecule that causes them to change shape. That's what allostery means, other shape. Allo is other, steri is shape. So there's a site in there where a molecule can bind. So here, for example, on the right side, that protein has bound to a small molecule, like an effector in this case. And that binding of the protein to that small molecule causes that protein to change shape. So in this drawing, in this cartoon, the regulatory protein couldn't bind to DNA without this small molecule bound because its DNA binding domain was in the wrong shape to actually physically interact with DNA. But when this protein is bound to that small molecule, this is where the sensing happens. This is the sensing of the environment. That small molecule, the effector, has something to do with sensing. So in the case of whether or not a bacterium is using a particular carbon source, that molecule is a sugar molecule, for example. So if it wants to sense what sugars do I have in my environment, the bacteria might have proteins that bind to those sugars and change the shape of the protein depending on whether the sugar is present or absent. So in this case, when the effector is present, this protein can bind to DNA. You can also have the opposite, where in the absence of that small molecule, the protein is normally bound to DNA, but when it binds the effector, the small molecule, then the protein falls off of the DNA. So same process, small molecule binds protein, causes protein to change shape, and that can have either effect. It can either cause that protein to be able to bind to DNA, or it can cause the protein to no longer be able to bind DNA. Look, that's what the next slide said. Any questions or concerns at this point? So we've got proteins that have evolved to be able to bind and interact with small molecules that have something to do with the environment that a bacterium is in. And those proteins change shape. When they're bound, that binding either causes the protein to be able to or not to be able to bind DNA. Now we get to leverage some of your knowledge about translation. All of the genes that are necessary for bacteria to use the sugar lactose are the three genes LacZ, LacY, and LacA. Yes. Stay healthy. It's not a good time of year to get sick. Not before finals. So LacZ, LacY, and LacA are three different genes that are used by these bacteria to metabolize the sugar lactose. So So they all need to be there present. The proteins that are encoded by those genes need to be in the bacterium if it's going to use lactose. And presumably that's why they're found in the bacterial genome as an operon, right? All the genes that are related to a similar function all found next to each other on the chromosome. And operons are transcribed as one unit, which you may have heard of in other classes. So you get one big messenger RNA molecule that has the transcriptional units for making those three different proteins. Z encodes beta-galactosidase, Y a protein called permease, and A encodes a protein called transacetylase. So you get one transcript, you get three proteins. One messenger RNA molecule and three different polypeptides. How does that work? Thinking back to translation. If you've got one messenger RNA molecule, how do you get three different polypeptides out of it through translation? Pardon? So no splicing in prokaryotes. That's the question. So this is prokaryotes. How does the ribosome associate with messenger RNA in prokaryotes? Shine-Dalgarno sequence. The ribosome binds a Shine-Dalgarno sequence, and then it translates until it hits ribosome sliding along the messenger RNA until it hits the first stop codon. So what exists in this operon messenger RNA that allows this messenger RNA to produce three different proteins? Yeah, three different Shine-Dalgarno sequences. So there'd be one there, and one there. So the ribosomes bind at three different spots on this messenger RNA molecule. There'd be a ribosome that binds there and translates LacZ. Another ribosome binds there and translates LacY until it hits a stop codon, and a third ribosome binds there and translates LacA. Just trying to link concepts together. I know you've probably all let all the translation stuff slide out of your memory already, but let's bring it back for a second. So when the bacterium senses lactose, it wants to make all of these proteins, LacZ, LacY, and LacA. The operon gets transcribed. So we're about to go through the details. Sir?
Is the lactose considered an effector?
We're about to see, but yes. So short answer, right. Lactose is what the bacterium is trying to sense. That's exactly the point. So if lactose is present in the medium, the bacteria detects the lactose, and that causes the Lac Operon to be turned on. So you produce the proteins that you can use to metabolize lactose like this. So one of those proteins, beta-galactosidase, the LacZ gene product, is what cleaves that two-ringed sugar molecule into two single-ringed sugar molecules. And these glucose and galactose are much easier for the bacteria to digest, to metabolize. So one of those three genes, LacZ, LacY, LacA, LacZ does this. It breaks the molecule into more simple sugars. Wouldn't you rather be out there? But I've got cartoons in here. Weird scientific cartoons. So yes, absolutely, lactose is the inducer in this case. So this weird-looking thing that's chewing on the DNA is supposed to be RNA polymerase, as the caption there says. So RNA polymerase grabs onto the RNA and it starts transcribing or saying, I guess. DNA is tasty? I don't know. Right. So this is where polymerase binds and it starts transcribing the operon. What happens when lactose isn't present? This ugly thing, the Lac Repressor protein, is normally bound to, what sequence do repressor proteins bind?
Operator, sorry.
Hello, operator, yeah. So the repressor protein is sitting on the DNA normally in the absence of lactose. The repressor protein is just sitting there on the DNA. And so in the bottom here, RNA polymerase is trying to move past the operator into the coding region But when lactose is present, the repressor blocks polymerase from actually transcribing those genes. This is in the absence of lactose, right? No sugar, you don't want those genes transcribed, the repressor is bound to the DNA.
You have enough glucose.
What happens when lactose is present? What changes? So the repressor's sitting there, and then when it binds lactose, that causes a change in the protein DNA binding domain. We could do some wacky jacks if anybody wants to. Right? So that's how this allosteric works in the case of lactose and Lac Repressor. Lac Repressor can either be bound to DNA or it can be bound to the effector, lactose. You can't do both things at the same time. When lactose is present, this means that what's the repressor doing? It's binding to lactose and it's not binding to the operator. So is transcription activated or repressed when lactose is present? It's turned on. The repressor is not bound to the operator, so RNA polymerase slides right through, transcribes the Operon, LacZ, LacY, LacA are produced.
And then once the protein is produced, it can break down the lactose that
Exactly. So there's feedback here. So as you say, once all of the lactose gets used up by the proteins that were just transcribed and translated, what happens to Lac Repressor?
the repressor rebinds.
Right. Once the lactose is used up, the lactose leaves the repressor protein. The repressor protein goes back and binds to the operator, turns off transcription of the genes that would use the lactose that's no longer there.
But since there's like multiple copies of this gene, it sort of slowly after the
It would take a while for this to happen because there are lots of copies of the repressor protein, presumably, in the cell.
Yeah.
And yes, yeah. But there's feedback.
Yeah.
So the cell can sense when lactose is there, and it also senses when lactose is gone. If you use the term sense loosely. Please.
Yeah, so a lot of the things are like the same between prokaryotes and eukaryotes. It is one of these things that translates over because like what happens to people that are lactose intolerant.
So does this have to do with lactose intolerance? Something I'll have to look into. But many of the same mechanisms that we're talking about here for prokaryotes do also work in eukaryotes. The principles still apply. In eukaryotes, transcription is still regulated by proteins binding to DNA and doing something to RNA polymerase or to other proteins to turn off or turn on transcription. I'll have to look into lactose tolerance. By the way, that reminds me, remember, you might not remember, on the last 2 days of the term, last 2 days of instruction, Monday and Wednesday the last week, I don't have anything on the syllabus particularly in mind, so if you want to learn anything specific about genetics that we haven't talked about through Thanksgiving, email me, tweet at me, whatever. Suggest a topic, and then those last 2 days, you tell me what to talk about, what to help you learn about, make some activities for you to do. So just let me know if you have those sorts of topics in mind. All right. So in the absence of lactose, repressor binds to the operator. This is a summary. Presence of lactose, that binding of lactose causes the repressor to fall off of the operator, and then you get transcription. Okay, so I did the hard work for you. I should have not given this to you, but I realized it after I posted the PDF, so too late. I made this wonderful summary chart of what's happening at each of the steps. So in a normal bacterium, wild type, what's happening in the absence of lactose in the presence of lactose? So there's a summary. Now we're going to start looking at, because it's genetics class, not cell biology class, we're going to start looking at mutations and seeing what those mutations, how those mutations affect the various steps of lactose regulation. So where could mutations happen that would affect this process? Let's brainstorm. So wild type, non-mutant, what we've covered to this point, that's how regulation of the Lac Operon works. What sort of mutations could we imagine? Where would they be that would screw with this process? What are the DNA elements involved that we've talked about so far? Okay, so we got the gene that encodes repressor, which is LacI, I think, if I remember correctly. The gene that encodes repressor. Which, oh man, must be Friday. I can't even spell repressor. What other DNA sequences we talked about that could randomly get mutated in bacteria? What does LacI interact with? The operator. Okay, so let's start with that. So we've got two different DNA sequences that are involved in the process of lactose regulation. We've got the protein that binds lactose, and we've got the DNA sequence that repressor binds to. So here's terminology for you. Constitutive mutation. So anytime we're talking about a constitutive mutation, that just means it's a mutation that causes something to happen without regulation. It's a process will happen all the time, no matter what. It's like this car recall that happened. This is probably a decade or so ago. I don't know if you remember this anymore, but there was a particular make and model of car where they designed the floor mat in such a way that sometimes your accelerator pedal would actually accidentally get stuck down because it got stuck underneath the floor mat. Yeah, bad design. That's a constitutive mutation. That mutant floor mat causes the vehicle to be unregulated, always on. The accelerator is always pedal to the metal. That's an analogy for a constitutive mutation. A DNA sequence change that causes a process to happen regardless of what's happening. That is, a bacterium is no longer sensing what's happening, it's just always transcribing the Lac Operon, regardless of whether lactose is there. So there are two mutations that we're going to look at briefly. One is called the OC mutation. That's operator constitutive, not Orange County. And the I- mutation. So both of these are constitutive, both two different types of mutations that both of them independently will cause constitutive expression of the Operon. Okay, so here's one. The OC mutation. What happens if you mutate the operator sequence? What effect does that have on transcription of the Lac Operon? So which protein is bound here? The operator binds Lac Repressor. Okay, so Lac Repressor recognizes a particular DNA sequence here, the operator. What happens if you change the operator sequence?
The repressor might never bind to it, so you'll always have transcription.
Okay, so if you change the sequence that repressor normally binds to, repressor no longer binds to the operator, then transcription's always on. The repressor is never sitting on the operator blocking RNA polymerase from transcribing the genes. So that's why this is a constitutive mutation, when the operator sequence mutates.
Can you also make like the change in the DNA so that the binding with the repressor is much stronger? So
That would be interesting.
you'll never have, you'll still have tad bit of transcription, but it'll never be enough to like reduce the lactose.
So the question is, could you have mutations also in the operator sequence that make Lac repressor bind it more tightly? What would that do to Yes, that can happen. What would that do to transcription? If the Lac repressor really, really, really tightly bound DNA at the operator, it would stop transcription. Right. So that would be the opposite effect. You have the always on, the constitutive mutations, and then you have another class of mutations, which we'll get to an example in just a minute. But that's one type of repressor mutation. Yeah, please.
That's good. Constitutive means that the mutation is happening in the specific DNA sequence.
Right, so there are mutant bacteria where if you look at bacteria that always produce Lac Operon transcripts, some of those bacterial strains have mutations specifically in this region. So that's how this region was identified. So you find bacterial mutants that don't regulate the expression of the Lac Operon. You sequence their DNA and all of it, lo and behold, you find, as is drawn here, a bunch of different, not all at once, usually one of these mutations in a strain, little slight changes to the operator sequence, but they're all of the mutations in the bacterial genome are confined to this one particular spot in strains that don't regulate Lac Operon transcription. Okay. So there's the table again. I've added OC, what's happening. So we have the two mutations that are constitutive. One's OC, one's LacI minus. So what sort of mutation to LacI, this is the gene that encodes Lac Repressor, how would you mutate the Lac Repressor protein to make transcription always occur? So you could mutate the place on Lac Repressor that recognizes a specific DNA sequence that targets Lac Repressor to someplace else in the bacterial genome. Okay. Let's see what happens in the IS mutations. So either you looked ahead or it's a good key. No, I don't know. I'm absolutely true though. So mutations where in the gene that encodes the repressor that changes its DNA binding domain make it no longer able to bind to the operator sequence. So it basically has the exact same effect as the OC mutation. You mutate the DNA sequence, the operator, or you mutate the protein that binds the operator. Either one has the same effect, right? They're partners. They work together, the operator and the repressor. If you change either one, you get the same phenotypic effect. That is, no repression of transcription. You get activation of transcription. So in this case, how do OC mutations and I minus mutations differ from wild type? Well, just look at the effect of transcription. Wild type, of course, when there's no lactose, there's no transcription. When there's lactose, there's transcription. These two mutants are constitutive. There is no regulation there. In both cases, transcription's occurring. Okay, the last line of the table is the opposite. What could we do to make no transcription either in the presence or the absence of lactose? So you've got the sugar there, and yet still you don't transcribe the gene. So that's called a superrepressor, IS. Superrepressor. You don't get transcription in any situation. Lactose present, lactose absent, no Lac Operon transcription. So, yeah.
Are you changing the part of the sequence of the repressor that doesn't have that allosteric site so that it's always bounded to the DNA?
Right, so this is how the superrepressor mutation works. So the way you get repression is you want that repressor bound to the DNA. How does the repressor let go and activate transcription? It binds to lactose, lets go of DNA. You'll have that image in your minds forever. One day I want to have genetics class, actually everybody come act out some, you can Google it. There's some great YouTube videos of classes acting out transcription and translation, and maybe you're fortunate that you're not in a class of mine where I decided to try that. Interpretive dance of genetics would be fun. So yes, sorry, getting distracted again, at least there's no bird. right? If you mutate the part of the Lac repressor protein that binds to lactose, what sort of mutation would you want that causes repression in the presence of lactose? So you mutate the repressor protein so that it no longer binds lactose. That's exactly what happens in the superrepressor mutations. So you get a mutation to the part of Lac repressor that binds lactose. You change the shape of that domain so it no longer interacts physically with that sugar molecule. And then once the repressor can never bind to that effector molecule, lactose, it never changes shape at its DNA binding surface. So it's just sitting there on the DNA. It doesn't recognize lactose anymore. You mutate the Lac repressor gene to change the protein domain of Lac repressor that binds to the effector. And that's, any questions? That's the story. So there are a lot of different types of mutations. One of the goals from this class, did we cover the objective slide? I don't remember seeing it, but one of the goals was to have you be able to predict whether or not transcription is happening in different situations based on different types of mutations. So that was the goal, the objective for this. On the final exam, I will ask you to, like we did on the previous exam, come up with a type of mutation not related to translation, but just where would you mutate a gene to cause a particular effect on transcription. So you want to be thinking about reminding yourself, how do superrepressors work? How do these different types of mutations actually affect protein-DNA interactions? Now in the last couple of minutes, I just want to see what sorts of calico cat situations we've got going on here. Let's see. Oh, here's one. Okay, so I just want to walk through one. We'll go through this again next class. So next class, what we're going to do at the start is we'll pick up the last couple of case studies for epigenetics before we keep moving on into transcriptional regulation. So the next story we're going to talk about for epigenetics, start of class next time, has to do with X chromosome inactivation. Here's, why is this representing random X chromosome inactivation? I'll look through the rest of your submissions later, but this is one example. Is this what you drew for random? If not, why not? What's random about it? So the X chromosome, what happens when one X chromosome is turned off? So we've got X1 and X2. X1 has a black pigmentation gene, X2 has an orange pigmentation gene. So we've got blotches of black fur, we've got blotches of orange fur. Do you agree it's random, or is it a non-random? The one on the top. That's what we see in tortoiseshell cats, calico cats, random blotches of color. What do you think, if I want to scroll down, what's that cat going to look like? Where non-random X chromosome inactivation happens. Mostly one color? So it's not, so what's the expectation of random? If you've got two choices and you're randomly inactivating one, if you look at a cat, how much of the cat should be orange? Yeah, over half. So that's kind of the definition for X chromosome inactivation. That's the definition of random. About half the cat turned off one X chromosome, about half of the cells in the cat turned off the other X chromosome. So you get about 50% orange, 50% black. So skewed or non-random X chromosome inactivation, this is the definition that we're going to pick up next time for class. Right, when it's not 50% one X chromosome turned off, 50% the other, when it looks something like this. And as we'll see next class, this has maybe something to do with homosexuality. whether or not you have skewed or random X chromosome inactivation, whether your mom has skewed or random X chromosome inactivation. So come back on Monday and we'll start off talking about that. Have fabulous weekends, please. Don't get sick.